Assalamu alaikum. We are starting oral pathology series. The first chapter of oral pathology is dental anomalies. For the understanding of dental anomalies, we must first understand the normal tooth development or odontogenesis. So, starting with the odontogenesis, Bismillah Rahman Rahim. The contents of odontogenesis, we will understand the definition, the initiation of tooth development, stages of development, apposition and maturation. So, let's begin. Odontogenesis. Odontogenesis is a complex process by which teeth form from embryonic cells, grow and erupt into the mouth. So actually these are embryonic cells which start proliferation and eventually erupt into the mouth. This radiograph shows all the three stages of tooth development. For human teeth to have healthy oral environment, all parts of the teeth must develop during appropriate stages of fetal development. So the primary teeth or deciduous teeth start to form between the 6th and 8th week of prenatal development, 6th or 8th week of prenatal development, while the permanent teeth begin to form in the 20th week, 20th week or actually the 4 to 5 months period. So how the tooth development is initiated. At the beginning of 6th week, the embryo stomodium or primitive mouth is lined by ectoderm. The outer part of the ectoderm gives rise to the oral epithelium. The oral epithelium initially consists of two horseshoe-shaped bands of tissues. These are horseshoe-shaped structures. Horseshoe-shaped tissue at the surface of stomodium, one for each. This is known as primary epithelial band. Primary epithelial band. At the same time, due to the forming oral epithelium, there is a type of mesenchyme originally from the ectoderm, ectomesenchyme, which is influenced by neural crest cells that may have migrated to the area. So let's understand this in a simple way. There is a primitive oral epithelium, and underlying there is ectomesenchyme. This primitive oral epithelium is actually derived from the ectoderm, while the ectomesenchyme this is derived from the neural crest cells. These craniofacial neural crest cells migrate from developing midbrain and first two rhombomeres into first brachial arch. So you can see this is the ectoderm and this is the mesenchyme, and these two ectoderm and mesenchyme instruct each other in sequential and reciprocal way to determine the formation and location of highly specialized teeth like incisors, molars, canines and premolars. During the later part of the 7th week, the oral epithelium grows deeper into the ectomesenchyme and is induced to produce a layer dental lamina. So in the at 7th week, the primary epithelial band forms outer vestibular lamina, and inner dental lamina. This vestibular lamina forms the vestibule, and this dental lamina forms the teeth. FGF and BMP, these are actually the signal proteins which are involved in the proliferation and invagination to form the placode, dental placode or dental bud. So this growth occurs in the developing jaw areas where the future curved dental arches of the primary dentition will form. The dental lamina then begins to form eventually in the midline of both arches and progresses posteriorly. These are the stages: lamina stage, bud stage, cave stage, early bell stage, late bell stage, adult tooth. Stages of tooth development. At the 8th week, dental lamina give rise to some 10 dental buds or nodules in each jaw, which invade into the mesenchyme and form primordia of ectodermal components. So you can see this is the dental bud which is invading into the mesenchyme and forming the dental bud. All the primary teeth and permanent molar arise direct from the dental lamina. So this is dental lamina. So all the this is you can see. So all the primary teeth and permanent molar arise from these region, while the permanent incisor, canine and permanent premolars arise from the this region. This is successional lamina, or this is also known as primary predecessors or secondary bud from the primary bud. The abnormalities of the bud stage include the congenital missing teeth and the supernumerary teeth. What is congenital missing teeth? It is the absence of dental lamina and supernumerary teeth, which is multiple dental lamina or multiple teeth are present. You can see this is multiple supernumerary teeth. and this is congenitally missing teeth. You can see here. Cape stage. At 9 to 10th week, the deep surface of the bud invaginates deep into the mesenchyme, resulting in the cape stage of tooth development. The cape consists of the outer dental epithelium and the inner dental epithelium and central core of loosely woven tissue. This is the outer dental epithelium and the inner dental epithelium, and this is stellate reticulum consists of these regions. The mesenchyme, this is the mesenchyme which will give rise to the dental papilla, which eventually will form the dentine and pulp of the future teeth. And the these are the enamel knots which are non-dividing cells will form the cusp tips. The abnormalities of the cave stage include the shaping, the fusion, and the gemination in this stage. At 11th week, the dental cape grows and indentation deepens. The tooth takes on the appearance of the bell. Actually, the central cells of the bud uh, proliferates slower compared to the peripheral cells. So it eventually forms an invagination, and this invagination takes on the appearance of the bell. Four structures are formed: the outer enamel epithelium, inner enamel epithelium, the stratum intermedium, and the cervical loop. This is outer enamel epithelium, this is inner enamel epithelium, stellate reticulum, and stratum intermedium. This stratum intermedium is formed between the inner enamel epithelium and the stellate reticulum. Dental lamina. Dental lamina disintegrates and will not join until the final eruption of the teeth. The histodifferentiation of Bell stage, the inner enamel epithelium form amyloblast, which will form enamel. The dental papilla form the odontoblast and will form the dentine. The core of the dental papilla will eventually form the pulp. Dental sac is the ectomesenchymal cells which surround the bud and the dental papilla. This is this is a sac of ectomesenchyme. Histodifferentiation. Differentiation, which is early bell stage, deepening of epithelial invagination and growth of its margin leading to assumption of the bell shape. The folding of enamel organ to cause different crown shape is shown to be due to differential rates of mitosis and differences in the cell differentiation time. The determination of crown shapes is under the control of genes and their signaling proteins and growth factors, signaling molecules which we studied the FGF and the VMPs. For differentiation, at 14th week, the outer enamel epithelium and inner enamel epithelium meet to form Herdwig epithelial root sheet. At cervical loop, Her stimulate the odontoblast to secrete dentine. This is cervical loop, this is inner enamel epithelium, this is outer enamel epithelium. Also during the 14th week, the stellate reticulum collapse and deflate. The outer enamel epithelium and inner enamel epithelium fuse and form the reduced enamel epithelium. Reduced enamel epithelium protects the teeth, form the junctional epithelium. Abnormalities in the bell stage include the macrodontia, microdontia, and the peg shade. Defect in the apposition involve the enamel hypoplasia and enamel pearls. Odontoblast, which secretes the collagen, will eventually form the enamel matrix. The calcification stage occurs after 14th week, and the abnormalities may include the fluorosis stain and tetracycline stain. Fluorosis stains are formed on the enamel, while the tetracycline stain appear on the dentine. You can see this is fluorosis stain, this is decalcification, this is Turner's hypoplasia, and this is fluorosis. Calcification starts at the cusp tips and it proceeds cervically. Primary teeth require 2 years to calcify, while the permanent teeth require 4 to 5 years. The dental follicle forms the bone, cementum, and pedial fibers. So these all structures are formed. We studied the tooth development a little brief so that we can understand the abnormalities related to the tooth. We will continue from here in the next lecture about the dental anomalies. There may be abnormality in the number of teeth, abnormality in the shape, abnormality in the size, and abnormality in the structure. Hope you like the video. If you like the video, kindly subscribe it and share it with your friends. Thank you very much. Allah Hafiz.